my path for your kind and nice introduction, and I also wanted to thank you for all the work that you are doing in this EAI Academy, and I think it's been a great success. Thank you. Well, I'm gonna talk today about the wildlife in the rocks and in the surface. Try to understand how two things. One is to understand how life can be spread in the universe in different planetary environments, and also we will see how this may affect, may condition the search for life in other planets. And because I guess that there are many PhD students here in the audience, I wanted to summarize a little bit what's the astrobiology and what's the main objective of the astrobiology. And it's just to remind everybody is we understand, or astrobiology understand life as a consequence of the evolution of the energy and the matter in the universe, and finding life in other planetary bodies is a logical consequences of this hypothesis for sure. So it's always a target, a key target, finding this life. So as you can imagine, many disciplines are involved in this big task for sure, to understand the key processes from the Big Bang to the formation of the first galaxies, the first elements, the first molecules, planets, how complex chemistry appear, and for sure at the end how all this complex chemistry organize and form life, and that is then distributed and spread in our case in the Earth. So but let's at the end somehow what we are trying to demonstrate somehow is if or the question under this is if carbon-based life is or the biochemistry based in carbon is really universal. So that's a key point too. So nowadays we know more than 5,000 exoplanets that has been already discovered. So there is 1,000 and much more for sure that will come in the coming year, but there will be perhaps millions of possibilities for having habitable conditions. Might be ocean walls, might be icy wall, oceanic walls, rocky ones. We might have habitable environments in the surface, in the soil, or in the surface of these planets. There might be free living or not, or settlers that live together, scavengers, or just adventurers that colonize the depth of these new worlds for us, for sure. But so we can imagine a lot of environments. Okay, but okay, let's stay first at our solar system and see what we can do in those planets where in fact we can reach and we can study in depth. So and the paradigmatic case is for sure Mars. We know that Mars has for many data in many missions that Mars has had water, enough water to cover big regions of the planet, and accounting for an ocean of nearly 140 meter depth, most of it in the northern hemisphere. And yeah, there was abundant water. Perhaps, who knows, life could have the opportunity to appear in this planet. But we also know from different data and missions, in that case from the MAVEN mission from NASA, that the Martian atmosphere has been lost in the past 4 billion. So it's practically with a rate of 100 grams per second. You can imagine that after billions of years, the only thing we have is the planet exposed to a high ionizing radiation and very low and extreme temperatures that make apparently a barren surface and a barren planet. But well, who knows? It's entire planets. We cannot just keep the first image we have. So let's follow the water and see or try to guess where this water where did this water go. This is the well, one of the models that they somehow envision how was the water history in the planet Mars. So from this ancient environment that you know, might have hydrothermal, subaquatic, subsurface, etc. For billions of years, this water might still kept under the surface under several centimeters or even meters of regolith. So this is important to understand this, and this is one of the key subjects for many research in Mars exploration. So, and we know, in fact, there is a lot of water. There's water that's from the Mars Odyssey gamma X-ray spectrometer. We can see, sorry, gamma ray spectrometer. We have it has been detected a huge amount of water underneath of the north, mainly in the northern hemisphere. And the NASA Phoenix lander confirmed that in fact the water ice was just a few centimeters below the regolith. Okay, so there is water on Mars, and it's clear that, well, still is there. Okay, and recently the Mars Express probe identified or detected the possible existence of brines, or even at least big extension of brines under the surface, around 1.5 kilometer depth. But who knows, might be other places in the planet that might have this composition too, or with more water available. So as we can say that there is water on Mars. If life had any chance to be there, perhaps it's still there. And the point is just to be able to find or its current presence, or just the remains of life from the past. So, but let's first understand what happened on Earth. Let's understand the only planet we know that is life, which is the Earth. And the point is, where and how is life distributed on Earth? And here, as we know that life is mostly in those parts, the upper part of the Earth for sure, the continental crust, the oceanic crust of the oceans for sure, and the lithosphere, mainly composed of rigid rocks in this interior of the Earth. So that means that life has to adapt to these conditions that are completely different from the one we are usually see on the surface. Just to make to understand in numbers, the biomass we have on Earth. We can see here in this work that, for example, plants account for most of the biomass on Earth. This is in gigatons of carbon. And we can see how Archaea and bacteria, that is prokaryotes, account for, not bad, it's up 14% of the total biomass, which is not bad. Just to make a comparison, you can see on the right what the animals account for in terms of biomass. So from this 14% of bacteria, most of it, you see here on the right, most of it, nearly 90% of, in general, prokaryote bacteria and archaea live in the deep subsurface, as you see on the left, that again account for around 15% of the total biomass, which is, well, is an important amount. So compared to the terrestrial or marine. So this is a huge amount of biomass that is mainly provided by prokaryotes. So, and one of the main characteristics of the deep subsurface is that the cell density tend to diminish to lower with depth. As you can see here in the x-axis, we have the cell density and the depth. There is a trend, and with, well, to diminish in this. All these spots correspond to different projects or different papers in the literature that reported certain cell density. Another thing we have to say here is sometimes it's very, very complicated to decipher which is the actual cell density, because the way of life in the subsurface, mostly they prefer to live in clusters, in groups that sometimes is very complicated to count and to say, okay, this cluster I found, it has 100, 200, or 1,000 cells. It's very complicated. So that's how these microbes, these microorganisms live in the rocks. So there's different ways to do that. You can see here just this grain of minerals where microbes can be living in different ways. They might be living forming microclusters, like you see in the right. They may have some cryptoendolytic inside the rocks way of life, some epilithic just on the surface. Whatever the way they are bind or attached to the grains, to the mineral grains, the most important thing here is that they like very much living together. So life are not usually microorganisms are not free like this one you see here, or planktonic cells like this you see in this part. They are mostly forming biofilms or clusters, because the biofilm or clusters provide a lot of things that are good for keeping, for protecting the populations. It's like they provide protection, they provide water, they provide nutrients, etc. So it's the preferred way of living in the subsurface. And we have an example here, one of the pictures obtained with one of our colleagues here. CAB is a wonderful biofilm obtained in the deep subsurface of Iberian pyrite belt, where we can see here cells stained in red, and a big carpet of polysaccharides, of exopolymeric material that has been stained with lectins that usually stain polysaccharides. So is the preferred way of living in the subsurface and inside the rock in general. Okay, so we started very, very early we were interested in understanding life in the subsurface and in the development of the science and technology for planetary exploration, particularly Mars. And we started 2 decades ago with the Marty project. It was a collaboration between NASA and CAB. Many people, many colleagues here at CAP participated in this project. And what we did in this, the idea was to, as I say, to understand life under the surface, to develop technology for drilling and life detection. And we performed two nearly 160 meter depth drillings, where we collected water drill, core drill, multiple analysis, geochemical, microbiological, etc. And the project consisted in testing. Well, I'm not going to talk here, but was the main objective of this project. Okay, here I can show you the main physical-chemical variables. In that case, geochemical variable we measure in these samples from 80 meter depth, where is the level where the water table was. So we identify the presence of a small organic acid for metal oxide that are usually good electron donors for microbiology, nitrate or nitrite, and sulfate that are also a good sector for electron change transfer, or the presence of hydrogen gas or methane gas that was detected in different part of this drill, including some part with abundant iron reduced iron or oxidized iron too. So one of the key points of life, as I said before, is very irregularly and heterogeneously distributed, and we identify, like in this case, small clusters of cells that were mainly associated with these browny yellow thin spots that we identified in the middle of the core samples, where you can see here was plenty of a lot of pyrite that you can see in this bright spot in the rock. Pyrite, the iron sulfide, is very is a mineral that can be used by many of the microbes living here as energy source, because they provide energy for metabolism just by capturing one electron in the reduced iron in the pyrite. So we also identified organic material is bound to the mineral, stick to the minerals. Mostly of this material for sure must be polymeric, perhaps polysaccharides, and when we are talking polysaccharides or extracellular substances, might be also some proteins, even DNA, whatever in this stuff. Or you can see here on the right how cells are even stick to the minerals. So we did a molecular characterization of all the drill cores down 80 meter depth. So we using different techniques. In that case, we use antibody. In that case, DNA macroarrays using specific probes for different phylogenetic groups of bacteria and archaea. We also some immunoassays, like I will talk to you later, and we also did DNA sequencing for phylogenetic studies. All these studies, we identify, for example, specific areas that were especially enriched in firmicutes, like bacillus stuffs that have relevant metabolisms, or we also identify some bacteria that are able to single carbon metabolisms, or we identify the presence of methanogens and methanotropes in different parts of these cores. So all together, we were able to make a summary of what were the main geomicrobiological processes that were operating in this part, where in the upper part, with a small microaerophilic environments, microbes like Gallionella and others were able to pyrite oxidation, even others of like Methylomonas from methanotrophy, and in a small, perhaps anaerobic patches might have some methanogens, nitrate reductions, etc., where they were used, perhaps using acetate or hydrogen as electron donors. So all these microbes were identified with different probes, and they might be able to perform these metabolisms. In this area between 120 and 150 meters, we identified some microbes that were able to, or at least are able to perform surface reduction, or even the consortium, the Amne, Archaea, and the sulfosalcina that can be able to couple anaerobic methane oxidation with sulfate reduction. So all these metabolisms can be there. The microbes were identified, and for sure it can be performed, this type of metabolisms. In the bottom part of the drill, we identified that there were some cracks where some water and even some oxygen coming into the substance. So in that case, we identify some microbes that were able to oxidize pyrite, while others were able to reduce iron, or even others may able to do methanogenesis, etc. So the point is that extracting the information from the rock with molecular techniques, we can infer a complex metabolism, metabolic interaction like this. So what we did is, there were many questions still to answer in this project, and we obtained a new one. In that case, was an advanced grants PI by Ricardo Mills, one of our colleagues here. It was an advanced grant from the ERC, and the idea was to go again deep, drill and go deeper in this project to answer some questions that still were remaining in the previous projects. Again, dealing with hardcore rocks is a complicated task. It's not easy, and we had to, we will see later, we had to deal with one critical problem, which is the contamination. But in any case, as you can see here, we had to use some tools that are most are properly from other disciplines that from biology, but we have to deal with these things. Again, we extracted samples and we did a geochemical analysis with depth. We selected from 100-meter depth because it was where 100 meter was between 90 and 100 meter was the water table, and we were sure that from 100 meter to the bottom we were in the anaerobic part of the drill. So, and you can see here we identified different compounds that like gases like hydrogen, methane. We also determined the total amount of sugars, proteins, even some the total organic carbon. We couldn't detect any specific pattern in this or in all these drill, but there was a kind of heterogeneous. Similarly, we did with the organic and inorganic anions, because they are very important for the inorganic anion as electron acceptor, the organic anion like acetate, formate, propionate, are very good electron acceptors for different metabolisms. So again, we have a distribution, which in some sample we have higher amounts than in other, but not any specific pattern that we were able to discriminate. So at the time of using molecular biology or molecular techniques in general with this type of samples, where the concentration of life is very, very low and very heterogeneous and very patchy, we have to be in mind to different things to deal with the signal to noise problems. You know? So we need to improve the signal in every step we take. So for increasing the signal, for example, in the DNA extraction, we have to improve as much as possible with doing different extraction, increasing the amount of sample, whatever. No? We also had very important to reduce the noise, minimizing the effect of contamination during DNA extraction. Just imagine you are dealing with sample with very little amount of DNA, and perhaps with very difficult to extract, because there are a lot of minerals that are interacting with DNA, and DNA love minerals and tends to bind to them. So we have to minimize this effect as much as possible. And whatever contamination from the outside, from the environment, may ruin your experiments. We have to minimize also the effect of sequencing error, because we are providing the system very little amount of DNA, and whatever mistake is also amplified. To pay attention to this too. And for sure, we have also to the post-sequencing processing of the sequences of analysis has to be also taken into account to avoid to eliminate whatever biases due to the amplification system we are using, because in all these projects we use, and perhaps in whatever project that try to study this type of sample, has to use multiple displacement amplification to amplify the amount of DNA you are extracted, because otherwise you practically have no enough amount for analysis. So having this in mind, contamination is always a nightmare. Contamination is always there, you know, and we have to deal in every step with it. So in the drilling operation, try to use, you know, all this system for drilling it, a refrigeration system. All of them use some liquids and some polymers that have to be used for keeping the system cool. So we have to be careful with that and using at least components that we can follow very easily. Okay, we have to be careful in the processing of the cores, in the sampling, the fine sampling atmosphere, where we have to take those part of the cores that we wanted. We also need to take the interior of the cores, where we can avoid as much as possible whatever external contamination. So in that case, we use a rotary hammer drilling, so that we can obtain cuttings from the inside, from the interior of the rocks. So, and always checking the temperature to be sure that we are not destroying something by using too much temperature. Further step, for molecular analysis is important. Once we obtain this powder, homogeneous powder, we distribute in different aliquots for DNA extraction, for lipid profiling, for immunoassay, for whatever. The point here is we have to be very careful with our consumables, with our tubes, with our hands, or whatever we can have around in the laboratory, because we have to be sure that all tubes are DNA and protein free, really. Okay, so what we did here was to treat with a part of acquiring good quality tubes. What we did was to treat with UV radiation to avoid as much as possible that whatever DNA that was contaminated was that were impossible to amplify. Okay, so again, all the reagents has to be treated somehow, because we are not sure. Nobody will assure you that DNA polymerase enzyme is totally free of DNA, or the nucleotides preparation is totally free of DNA, or even your water for diluting reagents are free of DNA. So again, we treated all these reagents as far as we can, for sure, as far as we could with UV radiation for sterilization. So in that condition, we applied the multiple displacement amplification with the P29 DNA polymerase, and that way we were able to obtain enough DNA for PCR amplification and 16S ribosomal RNA analysis, or for metagenomic analysis. And for example, here is just we determined the total amount of sugar and protein with depth. You have here on the horizontal axis, you see the different samples with depth, and the different concentration we obtain of total protein of sugar in micrograms per grams, I think. We also determined the taxonomic composition of the prokaryotic community, well, in that case, bacterial community, with some different samples. You see here with the number indicate the depth in meters, and other sample that we run as control, like drilling water, or another control from the exterior. So what we saw here is that what brought our attention was the special concentration of this green bar that correspond to Cyanobacteria sequences. So at the beginning we thought, okay, what Cyanobacteria? What the hell are Cyanobacteria doing here at that depth, 300-meter depth, or nearly 500, or for near 400? So at the beginning we thought that that could be a whatever contamination, but it didn't seem the case because all other analysis, like in that case, non-metric multidimensional analysis, show how the drilling water, for example, fall very apart of the main samples, where you can see here in this chart the light gray correspond to sequences that were associated with epilithic or dry or arid or permafrost environments. So what indicated that somehow these sequences are completely different from the one we can obtain from the drilling water or the outside of the area. So we also perform metagenome sequencing. One of the two of the samples were within enough DNA to make the whole metagenome sequences, and here you can see the phylogenetic association of the sequences. Most of them were associated to cyanobacteria, because we were interested in that sample where cyanobacteria was produced the highest amount of ribosomal RNA sequences. Here, where you can see a chart metabolisms, the metabolisms we identified within the metagenome. So a lot of them that account for a whole metagenome somehow. Okay. So interesting pathways in the degradation of lignin or aromatic compounds were identified. You can make this map. There's a great job that was done by Fernando, was one of the PhD student that did all this work. So all these pathways were identified in this metagenome at 420 meters. And in fact, yeah, the cyanobacteria were there. We identified later after multiple tests with fish and carfish of different samples. And in fact, here, what you can see here, there are this pinky stuff correspond to some of the cyanobacteria using different probes with the technique of carfish that stain the cyanobacteria and indicated that how they are still viable, because otherwise it couldn't be stained with the RNA probes. So yes, they are there, and we identify with other methods that we will show you later too. So, but how can a cyanobacteria live under in the dark and under these conditions? So, well, we identify in the metagenome the pathways and the enzymes that that provide these cyanobacteria the capability of obtaining energy by nitrate reduction or through electrogenic way here at the bottom, using always hydrogen as an electron donor. This is, as I mentioned before, the presence of hydrogen was abundant along the whole column of the drill, and this enzymatic system that can drive this energy through in that case, in the upper part, through the reduction of nitrate or through an electrogenic way that has been described in cyanobacteria. So altogether, allow us to make this picture with all those genes identified that encode for the proteins and the enzymatic system necessary for photosynthetic, that they still have this capability. One thing is they can do it, but we suppose that under this condition, no, but still keep the photosynthetic capability, fermentative processes, respiratory processes. So all these enzymatic system can drive the hydrogen. All this hydrogenase system can drive the hydrogen through the electron change transfer, and to either go through external electron acceptors that might be some phenolic compounds or some iron and manganese minerals, or using the nitrate as electron acceptor. So we also identify nitrogen fixation genes, Calvin gene, and the other nitrogen and iron sulfur assimilation system. So yes, we can say that this cyanobacteria might be viable there and might be using this system for life. Altogether, the data provide us a huge information of how life could be in the surface, with a very tight consortium of bacteria, where fungi can provide compounds by degrading the kerogen present in the rocks. The electrogenic cyanobacteria can provide some extracellular matrix and compound that can be used at the same time by aromatic degrading alpha proteobacteria, etc. So the point is that they all together might form a very tight syntrophic cluster that somehow remind us what is called our holobiont. Somehow it's a kind of, well, let's say, the starting of a primitive complex organization. But okay, just leave it as it is, because we have no more tests. But the idea is, okay, the fact that they live together, the environment, the rock and the deep surface environment somehow promotes this type of living, of very tight contact between different microorganisms. And in fact, we isolated and identified one of those microorganisms. This is what we call Tesserococcus lapidicaptus, which is captive of the rock. And it's a new strain of cyanobacteria that has a capability for nitrate reduction. Another environment that we study in the depth was the Salar Grande. Salar Grande is a big salar in Chile. It's a salar that has in some part 100-meter depth of pure halite. So it's that has been deposited depth from 9 to 7 million years ago. So it's a nice example to study, because there also, among other things, there have been discovered certain places on Mars that contain some halite deposits. So again, with these different drills, in that case, it was not so deep. It was a 5-meter depth drill, but I can show you here how the tools had to be used in that case, how we obtained the material from the interior of the core. And also very important is the liquid strengths in these environments in the Atacama, where halite crystals or perchlorate crystal of nitrate or other sulfate containing crystal acts as or has the capability for a trap water on his surface, producing the liquescence phenomena. And microbes only need a small thin of water, small thin films of water, where they can perform their metabolisms. In that case, we identify the presence of formate, hydrogen 2, acetate, propionate, that are electron donors for different sulfur-reducing bacteria, nitrate-reducing bacteria, or even perchlorate-reducing bacteria that can use or nitrate or perchlorate or sulfate as electron donors. So they don't need a huge amount of water, just a few bioavailable molecules. We studied the lipidomics and biomarker profiling with depth in another drill project in the Salar Grande, from 0 to 100 meters. Here in that part, we have two different units. The halite unit is down to 40 meters, and the detrital unit, which is down to 100 meters. So, well, here is just an example of different lipid biomarkers we identified, pristine, P10, and others. So we can see, for example, here that the halate layer contain the higher amount of this by unit of mass. So again, for example, here we compare to sample a 19-meter and 18-meter depth halate and detrital part, where we identify different opanes. Opanes are nice biomarkers that because they can they have a great stability and can survive somehow for even billions of years in geological records. So yes, they are there inside the salt. We also identified that in fact there are inclusions in the small halide crystals that you can see here, where some biological stuff is still preserved in the interior of these crystals. And even recently has been published a paper that just the beginning of May, a paper where they claim an 830 million year old microorganisms in primary fluids inclusion in halide in a sample. So, well, there is always a big controversial in this type of claiming, because it's very difficult to be sure that these microbes in fact has this the age they say they have. But okay, but it's interesting because fluid inclusion is a good way to preserve even not only organic matter but even, why not, living organisms. But okay, the point is, how can this work help us to search for life in other planetary bodies? Well, there is important point here is that the critical constraints that most of the instrumentation for planetary exploration has, and it is that we have to handle instruments that are millions of kilometers apart, and we have to simplify the procedure and protocol. So far, the only instrument that has been sent to Mars or even for other planets for detecting organic matter are based on the detection, are based on the detection of volatile compounds, and these volatile compounds are usually produced by pyrolysis or by heating, and this is a very inefficient way of extracting organic. Now, for example, the Perseverance rover has a Raman spectrometer, and it was foreseen for the ExoMars to have a laser desorption, another system that allow us to also to determine the presence of some organic matter just by hitting a laser on the samples. And what we propose for many years is the use of a bioaffinity system. In that case, is use an antibody microarray, where I would say the philosophy is the opposite. That instead of trying to remove the sample, the organic material from the minerals, what we just do is, okay, we approach to the mineral. What we do with this system is we homogenize a sample, just put into a liquid solution, and just let the antibodies go to the sample to fish somehow what are in this sample, that usually are nanoparticles or organomineral complexes. But that's our idea, and I explain it to you. The system consists of using a multi-antigenic test. We are now all familiar with the antigen test for COVID. So what we use here is not only one antibody, with 200 antibody or even more antibody that are interrogating for a similar number of targets. So then after a crude extract, we filtrate a little bit below 10 microns, incubate the microarrays, and then we add another fluorescent antibody mixture that if something has been captured, now these antibodies bind again to form a kind of sandwich that is revealed by using a laser that excites the fluorochrome, and a CCD camera take a picture of fluorescence. This fluorescence can be quantified, and the idea is to get direct information from the dirt to this data. Okay, so using this system, we have analyzed all the samples I've showed you before from the depth, from the Marte project, from the Rio Tinto down to 600 meters has been analyzed with this system. As you can see here, we identified many positive spots that correspond to, in that case, for gram-positive antigen, exopolymeric material, some proteins, etc. Or in that case, is a sample from the El Tatio. Recently, we analyzed samples obtained from a drill core on site, and we identified the presence of different microbes in this dust samples. This is the case from the Rio Tinto, where we identified for the first time the presence of Cyanobacteria. This antibody here was indicating that there were the presence of some biomarkers corresponding to the was very similar to the one we used to produce the antibodies, so revealing the presence of these microbes there. We did a total vertical profile of all the samples down to 600 meters. That you can see here, all this red stuff correspond to positive that I'm not going to enter into details because there's many things here to explain. But the point is there is a lot of organic, there are many microbial biomarkers and a lot of organic matter on the right that indicate that is a sample where life is present. Okay, this is similar to the Atacama. Well, I think it might running out of time, but okay, this is the Atacama where we did a whole profile with all the drill sample collected in the Atacama down to 90-meter depth. So you can see here all this bar corresponding to the quantification of the fluorescence of the chip, and all of them correspond to microbial marker. The green one corresponded to cyanobacteria that was identified down to 90-meter depth in the Salar Grande in the Atacama. So, well, the point is our intention is for sure to implement this system, this chip into the solid system, the cyanolite detector system. This is the sample preparation unit with its 10 extraction cells. On the right, the sample analysis unit with the laser, the chips, etc. And the idea with this is for sure to test. We have tested in many environments here on Earth. This is in the Arctic, where we identify many different signals and different samples, rocks around this area. And okay, the system works here on Earth, and it gave us a good results. But the idea is to implement this system for a mission to Mars, and we are collaborating with NASA Ames in their concept mixed mission, which is Icebreaker. The Icebreaker aims to send a mission to the Phoenix landed site or similar areas in the northern terrain because of the changes in the obliquity of the Mars during the last 10 million years that indicated that might have been suggested to many period of a great exposition to the higher insulation that might have favored the activation of the permafrost. So this is one of the targets, okay? And we've been working with them to mature the technology, to mature our light detector chip, to mature the solid instrument, to mature the drilling system, the robotic arm, all the entire platform for this mission. And in fact, I don't have here just a video to show you how it works, but it didn't work. Okay, doesn't matter. This is the last campaign we perform in the Atacama, where we perform a fully remote analysis using solid on top of this rover platform that we analyze completely different samples in a remote way, and we identify different positive spots, and we make some relevant discoveries about the biology in the sampling area. Okay, so just to say that very recently NASA has released the mission concept study for that is now in the Planetary Science and Astrobiological Survey, which is the Mars Life Explorer. The Mars Life Explorer, the target is again similar to the one we have in Icebreaker, is to target the permafrost now at mid-latitudes. And the idea is to use again the Phoenix lander and the icebreaker like drill, which is even mentioned in this document. So yeah, well, let's say that we are on the way, and that means that at least all the work we have done along these many years now seems to have its effect. And just to summarize some key points here is just okay to some messages to bring home is okay, life don't need oceans or rivers to thrive. We just need little bioavailable water. The liquescence phenomena are important, and I think will be very critical on Mars. Habitability can be extended well below the surface. So when we look for other planets, but I understand it's very difficult to explore or to infer the surface condition on other planets, but we have to have this in mind, not only searching for oceans. Rocks are good shelter for microbial life, for sure, and they prefer biofilms, the way of living. Minerals can provide protections, energy, and also bioavailable water. It's important. But one thing is important is that life is very irregularly distributed and has very low density, and sometimes it's very complicated to identify. And in that case, I wanted to mention that many instruments are just like Ramans, etc., has to be somehow improve the feasibility of getting or reaching the target by concentrated samples somehow. Biomarkers and cells can be very well preserved, which is a good thing in the rocks, or we need to make a new technological improvement to tackle with this. For finding biomarkers in these environments. And yes, for sure, we should go to explore the surface of Mars. Is clear. And just I want to acknowledge all this work. Most of the work shown here has been done during the Marte project and the IPVCL teams. And for sure, well, the solid team that is always behind this. Our funding agencies, the Spanish Ministry of Science and Defense, and CSIC, and for sure ESA, Astrobiología, and NASA, and DRC, and I don't know, perhaps there's someone else, but I think they are the most important. And just thank you to all of you for your attention. And I wanted to finish with this slide because as probably most of you know, is a picture from Mars taken by Curiosity, where we can see the moon and the Earth at the bottom. Thank you very much.